So let me just upload the PPT. Okay, you will recall that we had been talking about using the subject matter of biosensors, and very briefly before and this is where we had left off last time when we were discussing about how to use, for example, the lux operon, which gives fluorescence as a reporter system in order to detect the presence of heavy metals or any other pollutant. So the case study which I was discussing with you last time was for detection of naphthalene using the lux operon, and you also learned about what is the chemistry of the lux operon as to how bioluminescence is produced by the action of enzymes which are a part of the lux operon. So now, very quickly, so that is an optical biosensor. All right. So before I go to the next example of this, just revert back and recall that when you talk about, for example, the microbial biosensors, either we talked about the electrochemical transducers, where you had either amperometric or we had potentiometric detection, or optical transducers, where the signal between the analyte and the receptor is converted to something which is optical in nature. It could be luminescence, it could be fluorescence or something. And the third kind of a transducer, which can be used under microbial biosensor, is called as a microbial fuel cells. All right. So we had looked at the optical transducers, and before we leave off and talk about the microbial fuel cells, let me give you another example, which is for detection of mercury. So I'll go back to the slide. This is where we had left last time. Okay. And in this, I will very briefly remind you that, there are, for example, microorganisms which automatically develop a resistance to mercury. All right. So in a manner which is very analogous to the naphthalene operon, where there are genes which are located, which code for enzymes, which bring about degradation of naphthalene. All right. Now, in a similar fashion, if there are microorganisms which develop a tolerance towards mercuric ions, it is only because there is presence of an operon which is called as a mer operon. All right. So in a way, it is very similar to the naphthalene operon. That is, it is going to code for enzymes which are coded for by genes which are called as, like, MER stands for mercury. T is one gene in the operon, P is another gene, C is another gene, capital A and capital D. So all of these are actually genes, the products of which are responsible for detoxification of mercuric ions, which enters inside the cell. All right. And in a similar manner, as you learned with the naphthalene operon, there is also a segment which is shown in between here. All right. And this is a regulatory segment. And by regulatory, what we mean is that, this can be called as a promoter operator region, where the repressor protein or a combination of repressor protein plus mercury is going to bind. So this entire segment which you see here, for example, all right, is called as the operator promoter region. And to be very specific, when we say, for example, here, so T is the first gene, and when you say the promoter for T, this entire region is the promoter. All right, this is the -10 site you will recognize, because the RNA polymerases, they bind to a consensus sequence which is called as a -10. There is also a -35 region. So what is referred to as the promoter for T, that is because T is the first gene in this particular operon. All right, this is the -10, this is the -35. And in this particular segment, for example, the RNA polymerase is going to bind and it is going to transcribe the genes, the transcription of which is shown by this particular arrow. Okay, so the direction of transcription of this particular operon is shown by the black arrow. And at the same time, the segment which you see right here, all right, is called as the MER-R binding site. Now, MER-R is a gene which you see here. It codes for a repressor protein. Okay, so MER repressor, for example, has its own promoter, which is called as, like, this is the PR, which is the -10 and -35. All right, so RNA polymerase is going to bind to this segment, and then only the gene which is going to code for the mercury repressor protein is going to be transcribed in this direction. Okay, so like, for example, in any operon, the direction of transcription of these two functions, one is the MER operon, the other is the MER repressor protein. It occurs in the opposite direction. It doesn't have to be in the same direction on the DNA. So therefore, this particular site can be defined, because remember, operator is a site where the repressor protein is going to bind. All right, and since you do see promoters here, so this segment can be called as an operator and a promoter segment. All right, now how does it really function? When a microorganism becomes tolerant to mercuric ions, that means it is not, its biological activity of the microorganism is not going to be inhibited in the presence of mercuric ion, but rather it detoxifies. All right, so what is the meaning of detoxification here? In the case of naphthalene, detoxification means having the ability to break down naphthalene to salicylic acid, and that was one of the, not salicylic acid, it was another, the product of which, and finally it gets completely mineralized to carbon dioxide and water. In the case of mercury resistance, the meaning of detoxification is that the biological cells or any microorganism which you call as being mercury resistant is able to transport the mercuric ions and intracellularly reduce the mercuric to mercury, okay, which is like the vapor form of the mercury, and that is how detoxification occurs. In this process, you already saw that a number of genes are involved, which are coded for by either the MER operon or by the MER-R. Now, let's try to understand what is the function of each one of these. So MER-R, it is an exquisitely sensitive mercuric binding. In other words, this MER-R codes for a protein which is a repressor protein, which can bind to mercuric ion. Okay, and it is also a DNA binding protein. That is, when MER, either in the absence of mercury or in the presence of mercury, there is a change in the conformation of this repressor protein, and it is now able to bind to the DNA. Okay, as a result of which, this particular microbiological system gets primed for immediate transcription at the slightest influx of mercuric ions. Okay, now in order to understand what is happening here, you see the picture which I showed you last time is again shown here. RNA-AP refers to RNA polymerase, and these are the -10 and the -35 site. Now imagine MER-R is constitutively produced like most of the repressor proteins. Okay, so this protein, which is shown, you can just think that it is shown here as, for example, so it is a dimeric protein, and the protein, in the absence of mercury, all right, does bind to the DNA segment as is shown here. But then the conformation of the DNA is such that RNA polymerase can only bind to -35 and not to -10 site, as a result of which there is no transcription which is downstream. On the other hand, when there is mercuric ion present, okay, then the repressor protein can bind to this, and that is shown here. The black dot shows you the mercuric ion, in which case the conformation of this repressor protein changes in such a manner that it binds in a conformation now so that the RNA polymerase can also make a contact with the -10 site. So the difference between what you see here and the difference between this picture is that this is an inactive repressor, which is not able to cause transcription of the genes which are right here, but in the presence of mercuric ions, it changes the conformation and it activates this. So what you see here is actually denaturation of the double-stranded DNA and transcription. Okay. Now, once, for example, the transcription is turned on, then what all do you transcribe? You transcribed, for example, the gene products of TPC80. What exactly do these genes code for? Okay, MER-D, you see another one. This also has a regulatory function. Okay, MER-P, which you see here, or T or C. So all of these, they transport the mercuric species across the inner membrane. So in other words, the first step in mercury in mercuric ion detoxification is that the cells are able to take up the mercuric ions from outside, and that is because of the gene products of T, P, or C, which code for mercury transport. Okay, and then MER-A that you see here, for example, it causes reduction of mercuric ion to Hg0. Okay, and sometimes in many of the microbial systems, you don't have an MER-A, but rather you have a MER-B, which catalyzes the cleavage of carbon mercuric bonds in organomercurials. So, in the event that there are mercuric compounds which are organic in nature, that is, you don't have simply mercuric ions, but you could have organomercurials, in which case, for example, there is cleavage of the carbon and mercury bonds by an enzyme which is coded for by MER-B. Okay, so that is how it functions now. So the question is that, knowing this basic working of the mercuric operon, the question is that can it be coupled to the Lux operon? So Lux operon can now be used as a reporter gene, and what you would like to detect is the presence of mercuric ions in the medium. You want to develop a biosensor. In other words, and this biosensor, which is kind of a genetic circuit which you want to create, should be able to detect mercuric ion. And then what it should be able to do is that it should be able to translate the binding of mercuric ions into turning off the Lux operon. Okay, so you want to use Lux operon as a reporter gene. So that is what would be a microbial biosensor. So anytime there is mercuric ion which is in the medium, this microorganism, this recombinant system which you are going to create, should be able to simply light up, and that luminescence will be an indicator that there is mercuric ion present, which is in the medium surrounding this recombinant bacterium. So let us see how it can be done. Now, interestingly, apart from, look at the first picture here. Such a reporter system, it makes use of the operator and promoter mercuric, which is a DNA fragment, which senses the presence of the mercuric repressor and the presence of the mercuric ion. Okay, so I will just take you quickly back to the earlier slide. So in other words, this particular segment of the DNA, which contains both the promoter sequence and it also binds to this repressor protein, is now what we are talking about here. Okay, so this particular box which you see here is the mercuric operator promoter region, which can sense the presence of the mercuric repressor and inactive repressor or active repressor, in which case the repressor binds with the mercuric ion and it changes the conformation. Now downstream of that, what you have instead, you don't have something to detoxify mercury, but you have a reporter gene, which is now this luminescence, which is the Lux operon. Now, apart from Lux operon, you can also use the LAX-ZYA. And what would happen because of this is that the product of LAX-Z, again, you have the same sensing element like you have here, which will respond to the presence of mercuric ion. But then what does it turn on? It turns on the LAC operon, as a result of which you can metabolize, you can break down lactose by beta-galactosidase, or beta-galactosidase, which is coded for by LAX-Z, can also break down, which is IPTG, which is a colorless substrate, and it can convert it into a colored compound, which is blue in color. Okay, so in other words, if the cells are showing you blue color, that means that it can be that it's acting as a sensor. Presence of mercury is going to turn on the LAC operon, and beta-galactosidase is going to break down IPTG and give you blue color. That can serve. So all of these are therefore called as the reporter genes. Similarly, you have the green fluorescent protein, which is shown here. So instead of measuring luminescence, you could be looking at the fluorescence now, which is coded for by the GFP. In a similar manner, for example, there are a large number of other reporter genes which are shown here. All of these will belong to the category of optical based sensors. So you can have blue fluorescence protein, which is your reporter protein, and it can get turned on by the presence of mercury or by the presence of naphthalene, depending on what kind of regulatory and operator promoter regions you have hooked upstream of that protein. The output is going to be blue fluorescence. or you can have some carotenoid, some pigment formation, green fluorescence protein, GFP. I've already given you an example, and so many other reporter genes are there. Okay, and what is common to all of them is that they have a sensory element, which is so we have a sensory element here, which is going to sense the presence of either mercuric or any other heavy metal ion, depending on what this regulatory sequence is specific for. Okay, so I hope you're able to understand this. Now, this principle has been used in order to construct such a fusion plasmid in an E. coli system and create a recombinant E. coli which is going to sense the presence of mercuric ions, and it is linked to the Lux operon. It is not linked to the yeah, it is linked to the Lux operon, like you see here, for example. Okay, so these are all the constituents of the Lux operon, which I discussed with you earlier. All right, so now, what you see in this particular picture is, first of all, it is from a paper which is listed here. And the objectives which were set out in the paper were to construct a recombinant E. coli, which could be used as a sensor for sensing the presence of mercuric ions. And the response which is going to be recorded is that of luminescence, which is through the Lux operon. So the different constructs which you see here are, for example, so this is a plasmid which you see here. The Lux operon is right here, but upstream of the Lux operon, okay, you are going to put, for example, clone the elements which are the operator promoter regions, which are going to sense the presence of mercuric ions. Okay, R and T have the usual meaning. R stands for the repressor, okay, and then you have in between the repressor and T prime, okay, because remember T is the first gene. Let me take you quickly back to this particular slide. Okay, so what you are cloning is, for example, the segment of gene which is from MER, operator promoter region, and this MER-T, which is the first gene and which is responsible for transport of mercuric ions into the system. Okay, so this is what we have. So between EcoR1 and BAMH1, that is upstream of the Lux operon, it doesn't have its own promoter which is going to respond to, like you saw in the Lux operon. Normally there is that lactone which acts as a signal molecule, but it doesn't have its own promoter. Instead, the promoter operator region is going to be now this segment of DNA which is sensitive to the presence of mercuric ion. R gene codes for the repressor protein. T prime is the first one. This is exactly the same plasmid, except that the orientation is different here. Okay, you have a T, like you see here, you have a BAMH1 EcoR1, or if the fragment is reversed, it's in the other direction, because sometimes the response could be better in this particular construct. Then between BAMH1 and EcoR1, what is cloned are not only the first gene which takes up, okay, but the other genes which are a part of the mercuric operon. That means under this scenario, if you have all of these genes which you bring into this recombinant construct upstream of the Lux operon, since it has TP, it is it is able to transport mercuric ions inside the cell. And in this particular construct, which is POS 15, you see the entire mercuric operon, including the C, A, D, all the genes which are even going to code for reducing the mercuric ions into mercury, that entire mercury operon has been cloned. Okay, so these are, like, for example, in this case, this is only something which is, it's just a part of the T gene, not the entire T gene. So it is not able to actively transport mercuric. Okay. However, when you have TPC all together, you are able to take up mercuric ions inside. And when you have TPCAD, that means you are able to not only transport the mercuric ions inside the cell, but then you are also able to detoxify. You are able to reduce it. Okay. So that's what needs to be kept in mind, that all of these plasmids therefore will tell you as to what kind of gene constructs are inside. These are the kind of constructs which have been made, and then they have been introduced into an E. coli cell, which is going to act now as a microbial sensor. And now let us see what is the kind of a response which is recorded here. So first, let's see how these assays are carried out. Okay, so I think just let me make it a little bit smaller because it is overlapping with the written matter, and just give me a break here. Okay, so now let us see how these Merck and Lux assays are being carried out. So imagine now you have a recombinant cell, E. coli, which contains either the plasmid RB28, and I have already shown you the construction of PRB28. So PRB28, for example, doesn't contain, it's only a fragment of the T gene, not the entire T gene, but however it codes for the repressor. Okay, it also has the operator and promoter. Similarly, the POS14 and POS15, that is the response of POS14 and POS15. So there are three different plasmids now which have been introduced into the E. coli. So you have now three different types of cells, recombinant E. coli cells. Either they contain this plasmid, or they contain this, or they contain this plasmid. And now how do you really set up your assays? So the cells, the E. coli cells, which are now, for example, HMS 174, that is the E. coli strain, which carries the Mer-Lux plasmid PR828, or it carries POS14, or it carries POS15. And these are cultivated in two different media. Either they are cultivated in LB, like it shows here, for example, or they are cultivated in the minimal medium. Okay, so LB is a rich medium, minimal medium is just the basic salt medium. So let me just explain to you, for example. So in LB medium, so the closed circle here and the closed square here are, for example, the data which are obtained from this particular recombinant when it is cultivated in the LB medium. What is the difference between the circle and the square is that either, at 40 minutes. So closed circle means that the data is generated after 40 minutes, and the squares mean the data is generated at 100 minutes. Similarly, if it is cultivated in the minimal medium and you are taking your data, sampling is done at 40 minutes, you have open circle. If the sampling is done at 100 minutes, then you are going to have an open signal right here, like it says, and 100 minutes after addition of mercuric ions. Okay, so in this cell suspension which you have taken in a medium, gradually you are going to add mercuric ions, for example. The concentrations are going to be varied, and that is what is shown on the x-axis right here. So the numbers which you read here are 0.1, 1, 10, 100, etc. The same scale is used here. The same scale is used here. And, it says mercury concentration in nanomolar. And the same scale is also used for the other two data. So that's what we are trying to say here, that the cells are now the cell suspension you have got, and either you are cultivating them in the minimal medium or in the rich medium, that is LB medium, different concentrations of, so the data points which you are going to see here, for example, is the data which is obtained when only 1 nanomolar of mercury chloride has been added to it. And you are going to register your response for the cells cultivated in LB medium or in minimal medium, either after 40 minutes or after 100 minutes. Okay, so now what you need to see here is, for example, that so this is kind of the basal level, that is 0 level. Okay, so by 0 level, what we mean that there is some kind of a background fluorescence which occurs in these systems, because sometimes, like although we expect that the lux operon, is going to be turned on only when mercuric ion enters inside the cell. Okay, because then it's truly a reporter gene. But there is always a leaky expression. That is, transcription occurs along the lux operon, even in the absence of the mercuric ions. So sometimes this is a very common property in the biological system, that there is a leaky expression. Okay, so when you see a number here that is 0 mercury concentration, but still you are reading the number, and what do you have on Y scale, for example? Y is actually emission, which is the fluorescence emission, which is quanta per second per ml. So this is actually the luminescence emission, which has been measured in a measured in a luminometer, and that data is converted into quanta of light per second per ml. So in all the three cases, the response which you are looking at is recorded in terms of quanta per second per ml. Okay, so in this particular case, and all the details are provided to you here, how the Merlux assays are carried out. So read it carefully. It's also in the paper. And so that's what it is done. So in a system where you have only part of the T, which is it's not able to transport, as you increase the concentration, so quantitatively, as you increase the concentration of mercury ions, the response which you register is more and more and more. Okay, this response happens to be more on the minimal medium compared to that on the rich medium. On the LB medium, you see that the response is more on a complex medium. 100-minute data is worse. But at 40-minute, if you do sampling, it is able to register up to 1000 nanomolar. So, the sensitivity of this particular system, therefore, you can say that as you increase the concentration from 1 to 50, or for example, up to 50 nanomolar, right here, if you see here, okay, you are able to see an increasing emission of light in response to increase in the concentration of mercuric ions. Okay, so that is how it operates. Okay, so you see now that whatever response which you are getting here is directly proportional to the concentration, and that's exactly what you want a microbial biosensor to do. That is, as you increase the concentration of the pollutant, in this case it is the mercuric ion, the response in the luminescence is more and more and more. Okay, so you are able to quantify. Now, what we have in this picture, for example, is the data for POS14. Now, POS14, you will recall, is that it is able to transport the mercuric ions completely inside because it has all those genes in the construct which you have made. All right. So what you see here, therefore, this is behaving even more sensitively. That is, as soon, even less than 1 nanomolar, if the levels are like you see here, for example, maybe 0.8 nanomolar, or or 0.5 nanomolar, maybe immediately there is an increase in the fluorescence signal. But in this case, the signal is more responsive in the LB medium, and at 100 minutes, for example, you are able to register the response. So we say, and this is also hypersensitive, and the reason why it is called as hypersensitive is that in this particular construct, you are able to take up the mercuric ions, but you are not able to reduce it to mercury, to in the volatile form of mercury. You are not able to. So the cells become quite sensitive to mercury ion because they have not been able to detoxify mercury. Sure, they have been able to take up the mercuric ions inside the cell, but since you don't have that particular gene, okay, so in this construct, you will remember in POS14, you only have the transporter gene, but you don't have the gene A, which is responsible for making mercuric reductase. Okay, so the cells, once they take up, they are going to die. So it is called as a hypersensitive strain. So the second point, therefore, it indicates that the hypersensitive strain is responding to mercuric ion in the presence of 0.5 to 500 nanometer concentration range. Okay, so look at the sensitivities here. This is sensitive between 1 and 50, but the hypersensitive, really, its range increases quite a bit. You can go down even to detecting 0.5 nanomole, right up to 10 nanomolar range, you can detect, and the response is linear in this particular range. Okay, you can go up to 500 nanomolar, but there is a linear response up to this concentration. And finally, you have POS15, which also has the mercuric reductase. Okay, so in this particular case, you find that you are able to detect in this particular strain, because now it is able to detoxify it also, so the cells are able to withstand a lot higher concentration. So the actual response that you are going to register, the fluorescence, etc., is only operating after, let's say, 50, and it goes right up to this particular range. Okay, so it is just summarized here. that the lowest mercury concentrations tested which induced light were 500 nanomolar in LB and 25 nanomolar in minimal medium. Okay, so this way, in other words, what you have been able to construct is a microbial construct. E. coli, the lux operon doesn't belong to it, the mercury doesn't belong to it either. And both of these are taken from other microorganisms, but you have built up a genetic circuit which is able to respond to presence of mercury ions, and it turns on the lux operon. Okay, and finally, from the same paper, what has been done is that the data which is shown in this particular slide is the data which is generated in the laboratory, not with the real samples which are collected from lakes or from water bodies where there is mercury contamination. Okay, all of these are just recombinant cells which you are which you are taking in a test tube, and you are adding mercury ions from outside, and you are measuring the light emission. So now let me just read the legend to it. The rate of light emission by one of PRB28, again, it is that recombinant E. coli which contains this plasmid construct. And but the data in A represents freshwater data, which is open circle and which is closed circle. Okay, so these are the freshwater lake samples which have been taken, or rainwater, for example. So the samples which are taken here are, for example, the rainwater samples, and B is 1 to 3 dilution of an estuarine water sample. Okay, estuarine water sample, how is it really different? Because it has 30% salinity. Okay, so that is why it had to be diluted. Otherwise, too much of salt can also interfere with the luminescence output that you are looking at. So this estuarine sample, in other words, was diluted now. And of course, again, the closed triangles, the closed circles are, and the open circle here is a 40-minute data. And what you see here is the closed square, or the open square is a 100-minute data after induction. So it will be quite clear to you as you read the legend also. So two different water samples and two different sampling times. That is at 40 minutes and at 100 minutes. So again, what you see here, for example, is that, of course, it is spiked because in the freshwater sample also the level of mercury is very low, but these are the real samples which have been taken from outside, and some growth medium is added to it, and at the same time externally mercury ions. So in other words, if mercury is to enter into this freshwater body and you would like to measure, for example, how much mercury is present in there, you will take a sample of the fresh water, you will add a little bit of LB, okay, and you will add and depending on how much mercury is present, this is the kind of response which you expect to see. All right, so you see, for example, here that starting from 5 nanomolar concentration, there is fluorescence, which is now light emission there again up to this particular range. So in other words, this microbial biosensor which you have built up will be able to detect the presence of mercuric ions by virtue of the genetic circuit which you have introduced in it. And similarly here also, you will have the estuarine sample, which is from another water body. And if the mercury ion concentration in there happens to be this much, this is the kind of response which you expect to see. Okay, so that is really what the definition of a microbial biosensor is, that you are going to utilize using the recombinant DNA techniques in order to do this, in order to detect mercury. And in a similar manner, number of other metals, for example, or pollutants like naphthalene was one example which I gave you, or one could be looking at some other hydrocarbon, etc. One could be looking at some other metal like arsenic, chromium, etc. But provided you have sufficient knowledge about the kind of systems, about the kind of remediating systems which are present in the microbes, and you get hold of the repressors, corresponding repressors, which bind to those metal ions, or operator promoter segments which respond to the presence of those. If you have that kind of a knowledge, then you can bring up, it's a good challenge, good experiment. Sometimes you can just take it up as a project in one of the competitions to say that, well, look, I'm going to build up a sensor in this manner. And today, all that it needs is restriction enzymes, ligases, transformation, simple experiments, experimental techniques which you have already learned can be used to construct such kind of sensors. Now, so therefore, let me just summarize now. So eventually, when you start to build up a genetic circuitry like that, you enter into the field of synthetic biology. Okay, so for example, here is something which you will appreciate, that this is a microbial sensor based on synthetic biology. So what we expect here, for example, is input A. This input could be a metal or it could be some other pollutant which enters inside the cell. It binds to, the product of a regulatory gene right here. Okay, and the input along with the regulatory gene is going to bind to this operator promoter sequence right here. It turns on the transcription of a reporter gene, and then there is an output which is fluorescence, or which could be the green fluorescent protein. Okay, so you can imagine that this is a very simple, it's a single input, it's a modular biological pathway. It contains promoter and a downstream reporter gene. Okay, everything is contained within an expression vector. So this bacterial cell now, anytime it comes into contact with the signal, will give you, the signal is there and the output is there, which you are going to record. Okay, so that's what needs to be understood here. Or you can even build up, for example, something which is a little bit more complex. Now, this is the second example, which gives you something which is a multi-input device. So here, rather than only responding to one, there could be two different input signals. It could be something which you want to construct, which could detect the presence of arsenic as well as mercury. Okay, so you have, for example, input could be mercuric ion, it could be arsenic ion. These are the regulatory genes which are going to code for a repressor protein. They bind to them, and both of them, for example. Okay, so in other words, the promoter which you see upstream of a reporter gene, okay, now should be able to respond to the presence of both of these elements. That is, unless both of these products bind to it, it doesn't get turned on. Okay, in this case, it was a single input, but in this case, you design the sequence in such a manner that it requires both of these in order to turn it on. Okay, it could be some kind of a hybrid promoter which you are constructing. Like, for example, there is hybrid promoters which are built, are very, very common, which respond to the presence of two different elements. Okay, so you could have something like that, promoter operator, which responds to the presence of two input signals, and the output now is again luminescence or something like that. So this way, something like that can be built on a piece of paper and it can be put into action. So that is how optical sensors, for example, can be used. Now, the third example which I want to discuss with you amongst the microbial biosensors are what are called as the microbial fuel cells. Okay, they can work as a biosensor. Now, what do we mean by a microbial fuel cell? Okay, in a microbial fuel cell, number one, okay, what is important to know? This is like a two-chamber microbial fuel cell in which you see that there are two chambers here. In one, you have an anode. In the other one, you have a cathode. Okay, on the anode, what you have here is a group of microorganisms which look something like this. It is actually a biofilm in which there are bacteria which are embedded in here. So when we call it biofilm, that means you have the bacteria, but then they are all embedded in some extracellular polymer matrix. So they all remain clustered together when they give rise to a biofilm. Okay, so on the anode, there is a biofilm of these microorganisms, and they are not ordinary microorganisms. These microorganisms are the one which can metabolize organic compounds. Okay, now generally, whenever the microbes, they use an organic matter as a fuel source. Okay, they collect, organic matter is nothing but CHO. Okay, so what they do is that this organic matter is going to be oxidized to carbon dioxide. All right, all the hydrogens, remember that in the organic matter, CO goes away as carbon dioxide, and all the electrons or the hydrogens are internally within a microbial cell. They are accepted by NADH, or they are accepted by the various cytochromes, and oxygen comes in as a terminal electron acceptor. In these group of bacteria which you use in the microbial fuel cell, these bacteria, they externally donate these electrons to the anode. Okay, so there is no internal acceptor for these electrons. These electrons are thrown out into the medium and they are taken up by anode, where the flow of electrons will cause current to flow. Okay, the hydrogens which are now here, the hydrogens are going to flow through a semi-permeable membrane which divides the two chambers right here. So the protons are going to move to this side, and eventually, you do generate a current, and how much of current you're going to generate is going to be directly a function of how much of organic matter is metabolized by these cells, which are very special cells which don't internally recycle electrons, but rather they throw out these electrons. They are special group of bacteria. So how much of current is generated is directly dependent on how much of organic matter is there. So in other words, what you are going to use it is to detect organic matter. And this organic matter could be in the form of BOD, where a microbial fuel cell can be used as a BOD sensor, or it could be any other toxic organic compound. It could be naphthalene, or it could be some aromatic hydrocarbon compound, benzene, or it could be toluene, for example, which is an organic matter, which these microorganisms are capable of taking up, and they are capable of mineralizing it to carbon dioxide. The current flows, and eventually on the other side, the electrons and hydrogens will combine with molecular oxygen. So oxygen, you see, is entering in here, and it combines with the proton and the electron to give rise to water. Okay, so such a concept of a biosensor is called as a microbial fuel cell, and they are also being extensively looked at now. Okay, so some points I have just summarized here. So before we leave this topic, try to understand what we mean by microbial fuel cell, how it can be used as a biosensor. So you can use it as a BOD sensor, you can use it for detection of heavy metals. So microbial fuel cells are actually bioelectrochemical devices which produce electrical energy through the action of specific microbes. Okay, why do we say electrical energy? Because they make electricity flow right here. Okay, and sometimes the power is very, very dilute, but today people are trying to look at if these processes can be improved and if electric current can be generated using these kind of microbes. Okay, so in a typical two-chamber MFC, in a microbial fuel cell, like I said, the electrons which are generated right here are accepted, they are absorbed by the anode, they are transported to the cathode through an external circuit, and the proton exchange, like you see here, it occurs, and electric current produced by fuel oxidation can serve as a transducer. So that is how the microbial fuel cell. So let me quickly give you an example now here. All right, so here is a microbial fuel cell, a sensor for monitoring BOD, which has been developed. So what you see here, for example, is so in a microbial fuel cell, you have the organic water which is coming in, which is the wastewater, which has different levels of BOD. Okay, so biological oxygen demand, and the normal way of determining BOD is, there is a 5-day assay for doing BOD. In order to measure how much of BOD is present in the wastewater stream, it's a 5-day assay, and people have to be really very, very competent in order to do that BOD assay in the lab, because the results are often not reproducible, because it needs to be done very, very exactly. Okay, so imagine now that if you have a microbial sensor in the form of a microbial fuel cell to detect the presence of BOD, how, first of all, the time that it takes is very, very quick. You don't have to wait for 5 days to determine the BOD. All right, so the data is shown from that paper. All right, so the amount of current which flows. So in this particular case, for example, the BOD, so let me just expand it a little bit so that you can read the numbers a little better. Okay, so if you have, for example, 200 BOD, then the amount of current which is generated is so much, 0.38. If the BOD level is, which is about, I think, 500 plus, then the current which is generated is about 0.7. 300 BOD, or I think this is 200, 100 BOD, 450, 1300. So different wastewater streams have been supplied. Okay, and you see how much of current flows, and in this picture is shown that BOD, how much of BOD and what is the current response. So you see right up to about 700 or something, there is a linear response. So in other words, this particular sensor which has been built fuel cell sensor which produces a kind of a current, and on the anode there is a deposition of these special bacteria, and this microbial fuel cell is used for BOD sensing. You see now that the response is very linear. Okay, so now in summary, that microbial biosensors, so remember three types of biosensors which we talked about: the electrochemical, the optical sensors, and finally the microbial fuel cell. So they have been developed as a means for detection of a range of heavy metals, or like BOD, or it could be some other compound with increased specificity and sensitivity. The only thing is that there is a limitation using microbe, and that is, first of all, they become highly specific. Like one particular microbe, once you make a recombinant construct, becomes specific for detection of either only mercury or arsenic or something. So they're not as versatile under these conditions. And above a certain level, if the mercury concentration builds up to 1000 ppm or something, then it can also be toxic to the microbial system. Unlike a chemical system, if you have just collect a sample and measure it by GC or measure it by HPLC, you can measure very high concentration. But in a microbial sensor, beyond a certain level, they will not act as sensors, because that high level will be having a toxic effect. So with this, we end our discussion on microbial biosensors. So I'm going to stop here. And then when we come back and meet on Thursday, then we will take up the next topic. So that's it. So please listen to this lecture tomorrow at 9:30.